Hello, my name is Katherine Do, and welcome to the video for Module 2 of the Clinical Metaproteomics tutorial. In this video, we will cover peptide discovery. If you haven't already, please watch the two videos preceding this one, which are the overview video and the video for Module 1 database generation. These videos provide helpful background and context to understand what we are doing in this video. So let's begin. The agenda for this video will be as follows. First, we'll cover a refresher of the database generation module, which will lead us into discussing where we are in the Clinical Metaproteomics tutorial. Then, in the hands-on section, we will discuss the discovery module and start its workflow, which involves database searching using two peptide identification programs, managing their outputs, and compiling a list of confident, distinct microbial peptides. That will set us up for the next module, which will be peptide verification. Now let's do a quick review of the first module as a refresher. We started with our sample datasets that had been analyzed by LC-MS. Then, using three databases containing proteins from various species of interest, humans and contaminants, we created our large comprehensive database that we then reduced using MetaNovo. Lastly, we merged the compact MetaNovo generated database with the human Swissprot and contaminant databases, resulting in the input database for the discovery module. That is where we ended in the last video. Now let's see where we are in the Clinical Metaproteomics tutorial and what we are doing in the discovery module. So remember that the Clinical Metaproteomics tutorial is made up of 5 modules, each with its own workflow. In the previous video, we covered the first one, database generation. The focus of this video is the discovery module. Here we will use two peptide identification programs, SearchGUI PeptideShaker and MaxQuant, to identify peptide and protein sequences via sequence database searching. This will allow us to successfully retrieve reliable microbial peptides. This will be followed by the verification module, where we will use PepQuery2 to make sure that the peptides we've discovered are indeed of microbial origin and not misassigned as human peptides. Then, in the quantification module, we will quantify microbial and human peptides and proteins. And lastly, in the data interpretation module, we will perform taxonomic and functional annotation as well as statistical analysis. Next is the hands-on section. Similar to the previous video, I highly recommend having different tabs open for Galaxy EU and the Galaxy Training Network website. This hands-on section will include a brief setup that will lead into starting the workflow. And while the workflow is running, I will cover the inputs, the tools, and end on what outputs we expect to generate. Like in the first module, we will create a new history and import the datasets and workflow. I will quickly show these steps as they are the same as what we did in Module 1, but I do encourage pausing this video and try setting up the history with the inputs and the workflow on your own. The GTN website also has an FAQ section if you have further questions. So let's switch to the Galaxy EU website to set up. On the right-hand panel, click on the plus icon to make a new history. Then click on the pencil icon to name it. Here I will name mine CMP2_discovery. Again, I will also import the data using the links. I'm going to switch to the GTN tutorial page for this module, I will go down to the Data Upload section and copy the links in the black box by clicking on the button in the top right corner. Then I'm going to switch back to the Galaxy EU page, and on the left-hand panel, I'm going to click Upload, and then Paste and Fetch Data. I will paste the links into the text field, and then click Start. Once all of our data is uploaded, we can click Close. And again, we will create a dataset collection of our sample data to keep things organized. We have provided the same 4 example datasets that were analyzed by LC-MS-MS, with the only difference being that they are in RAW files to be compatible with software in this module. To create a dataset collection, first I'm going to click on the check mark icon in the History panel. I'm going to click all 4 example datasets, and then I'm going to click on the button that says how many datasets I've collected, and then Build Dataset List. I'm going to name this collection Raw Data Collection, and then click Create Collection. Again, I'm going to click on the checkmark icon to deselect my datasets. Next is getting the workflow. I've included the workflow link on this slide, and it is also on the GTN tutorial page for this module. Similarly to the first module, you can open this link in your web browser, click on the Import button, and start using it right away. Click on the Play button, and now we are ready to put in our inputs. We will start running the workflow and discuss the datasets and tools while it runs. We set up the workflow so that you just have to match the input datasets to the input name listed. Once you've done that, we can click Run Workflow. We will now cover the inputs and main workflow steps. We will end on the final expected output for this module, which will set us up for Module 3 verification. For tool specifics, please refer to the GTN tutorial page. Let's cover inputs. The input protein sequence database that will be used for database searching is from Module 1. Remember that this database was generated by merging and filtering the MetaNovo generated database with human Swiss prot and contaminant proteins. We have also provided the same 4 example datasets that were analyzed by LC-MS-MS. Remember that we just put these into a dataset collection in our Galaxy history. The only difference from the first module is that these datasets are in RAW format. The different file format is due to the fact that MaxQuant can use RAW files, but SearchGUI must have them in MGF format. You don't need to convert these files yourself, as we have included a tool in our workflow called MS-Convert that will convert from RAW to MGF. We have also provided a file called Experimental Design. This file is specific to MaxQuant, and we will mention it again in a few slides. It essentially tells MaxQuant how to correctly organize and analyze the input mass spectrometry data. Now let's cover what we are doing for peptide discovery. Here is the overview of the discovery module. As I mentioned on the last slide, inputs for database searching will be the reduced MetaNovo database from Module 1 and our sample MS datasets. We will use 2 peptide identification programs to identify peptide and protein sequences via sequence database searching. By using 2 programs, we will increase the range of detectable peptides. These 2 programs are SearchGUI PeptideShaker and MaxQuant. SearchGUI and PeptideShaker are separate software, but we will consider them as one program since we use them in combination. So, what exactly is database searching? Database searching uses mass spectrometry data to identify proteins. To do this, the search algorithms of these programs identify peptides in the MS spectra and search the input protein sequence database to match observed peptide data with the theoretical peptide masses and spectra. Outputs from these two programs will be filtered to select for confident microbial peptides. Ultimately, we will create one single list of unique microbial peptides. Now let's talk tools. First up is SearchGUI PeptideShaker. Remember that the MS Convert tool will convert the sample data from raw to MGF for software compatibility with SearchGUI. SearchGUI comprises different search engines to match sample MS spectra to known peptide sequences. In our analysis, we will use search algorithms X-Tandem and MSGF Plus within SearchGUI for matching spectra from our sample MS data against peptides from the protein sequence database that we acquired in Module 1. Additionally, the SearchGUI tool will require a parameters file. SearchGUI will not generate this file itself. We have included a tool called Identification Parameters in our workflow that will create this file for us. The SearchGUI tool will perform a database search based on this parameters file and will generate a SearchGUI archive file that will serve as the input for the PeptideShaker tool. The SearchGUI archive file contains peptide spectral matches, or PSMs, and to assess the confidence of these data, we will use PeptideShaker, a post-processing software. PeptideShaker also infers the identities of proteins based on the matched peptide sequences, and users are able to visualize these outputs to interpret results. PeptideShaker will generate various reports that serve different purposes in proteomic data analysis. For tool specifics, please refer to the GTN tutorial page. The other tool for database searching is MaxQuant. As I previously mentioned, we have provided an input tabular file called experimental design. Similar to the parameters file used for SearchGUI, the experimental design file is specific to MaxQuant and contains experimental conditions, sample groups, and relationships between samples in a proteomics experiment. This file helps MaxQuant correctly organize and analyze this mass spectrometry data. More details about this file can be found on the GTN page for the Discovery module. So, how does MaxQuant work? MaxQuant is a mass spectrometry-based proteomics platform that is capable of processing raw data, meaning we do not need to convert the raw data to MGF like we did for SearchGUI. So, in our analysis, raw MS spectra will be searched against the protein sequence database. MaxQuant also provides improved mass precision and high precursor mass accuracy. This results in increased protein identification and more in-depth proteomic analysis. MaxQuant outputs will include protein groups and peptides, as well as other files that will give information about identified peptides. Next, we want to manage our outputs to prepare for verification in the next module. To do this, we will use various text manipulation tools. More details can be found on the GTN page for this module. Some examples of text manipulation tools include the Select tool, since we only want microbial matches. We also have the Filter tool and the Query Tabular tool. These tools help us select confident PSM matches and query for their protein accession numbers. The Cut tool allows us to select columns, and the Group tool allows us to select unique entries, in this case, unique peptides. These tools enable us to obtain a list of unique, confident microbial peptides from both programs. This brings us to the final processing steps, where we will concatenate or combine the two peptide lists from SearchGUI PeptideShaker and MaxQuant into a single dataset, which we will group one last time to remove duplicates. This will give us the last output for this module, a list of confident, distinct microbial peptides. At the time of this video, we obtained around 196 peptides from this step. We will use this list for the next module for peptide verification. This marks the end of Module 2 Discovery of the Clinical Metaproteomics tutorial. To navigate to Module 3, go to the bottom of the Discovery tutorial until you reach the Go Further box. You can click on the link to continue with the next module. Please feel free to leave feedback about what we liked and how we could improve. We hope to see you in the next video, where we will cover Module 3 Verification.